All right, our last section for the cell respiration unit is on fermentation. And so we're going to kind of compare and contrast here the anaerobic versus the aerobic forms of producing ATP. And we're going to focus on the anaerobic side today. So when we talk about fermentation, it is an anaerobic process. That means when oxygen is not available for cell respiration, then we're going to go into fermentation. Okay? But one thing I want to note is that fermentation by itself does not produce ATP. No ATP produced in fermentation. But it does follow glycolysis. And if you recall, glycolysis produced a net of 2 ATP. So what fermentation does is it's an anaerobic process that allows glycolysis to continue, because the only way some organisms will ever get any ATP is if they do glycolysis and get those 2 ATP. There are some organisms like bacteria that only live in anaerobic environments, and so they need that 2 ATP. And so fermentation allows glycolysis to continue so that those organisms can get those 2 ATP. So again, all of our processes start with glycolysis, where we break down the glucose. If there's oxygen present, we'll go into cell respiration, and if there's no oxygen, we'll go into fermentation. Now there's 2 main types of fermentation. Lactic acid fermentation occurs in muscle cells and in bacteria. Okay. Glycolysis splits the glucose into 2 pyruvate, so we still have to have that happen first. Then the pyruvate and the NADH that are formed are going to enter fermentation. So if you look down here, we've kind of abbreviated glycolysis here. We haven't shown all of the, because remember, you still have to use 2 ATP to break down the glucose, and then we produce 4 ATP in the end, but so we get a net gain of 2. So that's what we're showing here. We're taking the glucose, breaking it down to 2 pyruvate. You get a net gain of 2 ATP and 2 NADH, as well as the 2 pyruvate. Okay? Then that pyruvate and the NADH go into fermentation. In this case, it's going to be lactic acid fermentation. The energy from the NADH, because remember, NADH is an energy carrier, converts the pyruvate into lactic acid. So here we're using this energy right here, and we're converting the 2 pyruvate into 2 lactic acid. Lactic acid is still a 3-carbon molecule. The NADH gets changed back into NAD+ when that energy is used, and then that NADH gets recycled back to glycolysis. So you can see here, here's the NADH, that's going to come back here and be used in glycolysis. So that's what we mean when we say fermentation allows glycolysis to continue, because when we're breaking down glucose, we're going to need these electron acceptors in order to make the NADH here. Now normally, if we were carrying on cell respiration, these electron acceptors would be coming from the electron transport chain. Remember when we bring all those electron carriers down there and they release their protons, those hydrogen ions, and their electrons into the electron transport chain? Well, then they convert back to NAD+ and FADH, FAD2+. So for aerobic respiration, we're getting all those from the electron transport chain, and some of those will come back to glycolysis to be able to start glycolysis. But for fermentation, we get them right here, and we have to recycle them back. So fermentation allows glycolysis to continue when there's no oxygen present. The other type of fermentation is alcoholic fermentation. This occurs in both yeast and bacteria. Again, the glycolysis splits the glucose into both 2 pyruvate, your 2 ATP net, and then 2 NADH. So we're still doing that step. This time, the energy from NADH splits the pyruvate into alcohol and carbon dioxide. So this time we have 2 different end products here. Once again, that NADH gets changed back into NAD+, and the NAD+ gets recycled back to glycolysis. Now, obviously, the alcohol is important in the brewing industry. You know, any Anheuser-Busch and some of those places that like to produce beer and other alcoholic beverages, they're utilizing this part of the alcoholic fermentation. But the baking industry is using the end product of carbon dioxide, okay? Because that carbon dioxide, when it's trapped in the dough and as it bakes, that's what's going to make your bread nice and fluffy. But the little bit of alcohol that's produced in that short amount of fermentation time, as the bread bakes, that deteriorates. So it's not like we still have alcohol in the bread. Otherwise, you'd be in trouble if you ate sandwiches, right? All right. So fermentation is used in food production. Both, if we go back here, lactic acid fermentation, remember I said it occurs in bacteria. Things like yogurt and cheese utilize lactic acid fermentation. That's why that yogurt, especially if you've ever had plain yogurt, it's very sour. It's like sour cream. It's very tart because it's released this lactic acid here. Okay? So fermentation can be used to make yogurts, cheese, bread. We already talked about the alcoholic beverages, but also things like sauerkraut. Some of you may not like that, but I like it on my brats in the summertime when I'm grilling. Also kimchi, if you've ever had that. That's like a fermented cabbage as well. So we use fermentation to make lots of different food products. So this last part is just to give you a little bit of practice, so you can always come back to this and erase it and try it again, just to review before a quiz or a test. So let's see again, this 6 carbon molecule here, that would be glucose. And we need 2 ADP, again this is net, to produce 2 ATP. And then we have 2 NAD+, which gets converted to the energy carrier 2 NADH. But our goal is to break that glucose down into 2-pyruvate. Okay? Then we're going to use that energy from the NADH to break the pyruvate down into lactic acid. And then that NAD+ gets converted back here to glycolysis. It gets recycled back so that we can keep that cycle going. Now, if the end products, if this was alcoholic fermentation, the end products would be alcohol and carbon dioxide. So you get 2 end products for that one. And again, what gets released in fermentation that gets recycled back to glycolysis, that would be NAD+. So that's why fermentation allows glycolysis to continue, is because of that recycling back of that electron acceptor. All right, so that's fermentation.